It's my pleasure to welcome all of you in TIGS webinar on hematological disorders, therapeutic and disease modeling. So, rare genetic disorders is an important health issue in the country. I'm happy that more and more awareness is coming for this. At TIGS, we are involved in this direction, along with our other programs on infectious diseases and crop improvement. And our focus at TIGS is essentially to, in the Indian perspective, because the number here is, although it's called rare genetic disease, the numbers are quite large. So therefore, we need diagnostics and other things which are high throughput and affordable uh, for the large numbers, and it requires a lot of R&D efforts, innovations, and of course the social awareness for this particular kind of diseases. As many of us know that inherited hematological disorders are among the most prevalent genetic disorders in India, with the hemoglobinopathies like sickle cell anemia having carrier frequency of 35%, and therefore the huge number of patients who require timely clinical interventions and management. While there are many other diseases like DBA or CDA, which are not well understood, and a lot of basic research needs to be carried out to be in a position to do something about it. So this webinar is focusing on a range of topics in sickle cell disease and how iPSC models can be used to study disease pathogenesis associated with inherited bone marrow failures. As I mentioned, at TIGS, we are working on gene editing based iPSC models for rare genetic disorders, as well as stem cell based therapeutics for hemoglobinopathies. In addition, we are trying to make diagnostics of various rare genetic disorders affordable in our country. I'm certain that this webinar will help us gain both insights on clinical practice and cutting edge advantage on the research in the field of blood disorders. We have very uh, eminent speakers, two speakers for today's webinar. And I will first give very brief introduction of Dr. Dipty Jain, who's going to uh, talk about sickle cell anemia in India, a clinician's perspective. And then second presentation will be by Dr. R.V. Shaji, who will speak about stem cell models to study hematological disorders. So Dr. Dipty Jain is a pediatric clinician by training and completed her MBBS in 1979 and MD in 1984 from the Government Medical College, Nagpur. She also did her master's in science in clinical epidemiology from McMaster University in Canada in 92 with a focus on health economics. She has been a professor since then at Department of Paediatrics, Government Medical College and Hospital in Nagpur. And currently she is the nodal officer for the Sickle Cell Project of CSIR and has served as a key member of various committees constituted by Government of India and the international agencies like WHO on sickle cell epidemiology. I had the privilege of interacting with her several years ago when we were running the first phase of our sickle cell anemia project, and I was so impressed by her attachment to the work that she's doing and attachment to the patient for whom she was working. I think she's among those rare gems we have in medical community who cares about to this level to the patients who are otherwise very underprivileged and who need help because there is not very straightforward treatment. A lot of attention is required, and she also contributes by several ways in the research that is going on in the country in this field. So we look forward to hear your presentation, madam. You can start this.
I wanted to thank you for the kind words and also for inviting me for this seminar. And you've chosen a very important topic, that is sickle cell anemia, which was quite neglected for some time. Next slide, please. And it's a hard task for me to cover this topic in 20 minutes. So I think I'll be very happy to answer the questions. If you have any, I may miss few things. Now, if you look at the global burden, it's estimated that Nigeria, DR Congo, and India, these three countries have the highest burden of sickle cell disease in world. Next, please. Now, if you look at the Indian epidemiology, um, I see there has been a changing epidemiology in India. Why I say changing epidemiology is because when we started, I knew it was West Gujarat, Central India, that is Vidarbha, from where I come, Madhya Pradesh, Chhattisgarh, Orissa, and some part in the south, the Nilgiri Hills. These were the states which had high prevalence of sickle cell disease. However, you can now see that Rajasthan, um, uh, and Kerala and other states have also joined as high burden of sickle cell disease. And there is a changing epidemiology in the last 10 years. It was not that they were not there, but I think the awareness and the screening has brought to our notice that sickle cell disease does exist everywhere, and we are still finding new areas where sickle cell disease. Point to notice, it is much less seen in the metros, Delhi, Bombay, Chennai, and Calcutta. And other thing is, it is thought to be a disease of tribal population. No, but that's not true. It is equally common in non-tribal population also, and now seen in all types of population in India. The other thing is the presentation of sickle cell disease, the clinical presentation is very heterogeneous with regards to the type of presentation and also the severity. If you go to Gujarat, the presentation would be milder than what we see in Central India. So that is what I mean by heterogeneity. Next, please. Next slide. A word about the molecular part of sickle cell disease, and we know there are different haplotypes. And what we have in India, which has been reported, and even we have found in our newborn screening, is Arab Indian is the majority of the patients have Arab Indian haplotype. Arab Indian is associated with high hemoglobin F, and it is around the mean hemoglobin F level in our patients is about 15 to 22% in sickle cell patients. And other thing is there is associated deletion alpha thalassemia. Sorry, it's not A, it's alpha thalassemia, which varies from 24 to 97%. 97% in the tribals and 24% in the non-tribals. This is our experience from Maharashtra. Next, please. Now, if I go through the journey so far, it was in 1952 that Lashman and Kadbush first described sickle cell disease in India, though it was described globally in 1910. However, in India, it was in 1952, first described in tribal population of Nilgiri Hills. In 1956, it was Solanki, who was one of the faculty at our medical college. He described sickle cell disease in a non-tribal population in a place called as Chandrapur near India. From 1956 to 1983, there was not much done in India about sickle cell disease. And in 1983, when probably I joined my pediatric training, I saw that I think 40% of my patients were of sickle cell disease in the ward, and it has very high morbidity in the form very painful, very pain. The child would be just crying, and that is why in African it is called as chi chui tui, which represents a cry of a child. That time I had nothing to offer in 1983, nor did I know much about it. So it was just folic acid and oral rehydration solution, Paediatrics, which I gave them, which brought the patients to me. And historically, I used to think it is a disease of childhood and they do not survive beyond the first decade. This was my conception at that time. Then in 1992, it was, we thought that we knew from other countries that penicillin prophylaxis does help in decreasing the infection. And till now, we were using paper electrophoresis. We switched over to cellulose acetate. In 1998 was, I think, milestone for us because ICMR conducted a study, a Jai Vigyan project, where 5 tribal population, primitive tribal populations were screened and managed for sickle cell disease. And I think here we came up with a test called as solubility test, which we now use as a screening test. So it was an important milestone. In 2000, it was Nagpur was the place where we started newborn screening and started an RCT on hydroxyurea, which is published. It was not till 2002 that I had a comprehensive healthcare. Imagine, I did NBS, but I did not put them into, I just wanted to know that the test used for newborn screening is good and valid. So it was only in 2002 I put them on comprehensive healthcare. We started prenatal diagnosis in 2002. Now, it was a stage of rapid progression from 2002 to 2016, what we have got today. And I think I'll be sharing that in the next slides. Next, please. Okay, now these are few of the clinical manifestations which we see commonly. The triad of sickle cell presentation in our country is pain, anemia, and infection. Most of them, majority of them will have that. They overlap their presentation, but these are not the only presentations. There are many other presentations, and the clinical symptoms change with the age of the patient. Like what you see here in the dactylitis in a young child. The site of pain are fingers in children below 2 years. Then the site shifts to knee and larger joints, and in older patients, it is ribs and the spine. Other is jaundice, severe anemia. Here is one thing where you can see that patients' parents are taught to measure spleen to prevent splenic sequestration, not prevent, early detection of splenic sequestration, which is a very high cause of mortality in children. Next, please. Now, because we have high hemoglobin F level, it was thought that Indian sickle cell disease is benign as compared to other population in US and in Africa, in Europe. However, this is not so. And here we have compared with the cooperative sickle cell study, which is an historical study, a cohort going on since 1982, from which we have learned many lessons from in US about penicillin prophylaxis, hydroxyurea, and many more things. But here you can see that India, except for leg ulcer and priapism, rest of the manifestations are much more than what we see in other populations. Next, please. I want to say that Indian sickle cell disease is different, and we do not know much about it. The natural history of sickle cell disease is not very well known, which can be done only by newborn screening. Now, coming to some challenges in the diagnostic test, as I said, initially we did a sickling test, which was a slide test where we would look for sickle cells, and then repeat, see the same slide after 24 hours and say, yes, this child can be sickle. That could be a trait or a homozygous. But then next came the solubility test, where we did not wait for 24 hours. Within 10 minutes, it was a 20 rupee test. In the field itself, we could say that the patient has sickle cell, but we cannot again say whether it was trait or a homozygous. But now we have point of care test, where I think 3 tests are very well available and they are very cheap, do not need battery or anything, and can be used even in newborn. This test, solubility and sickle test, could not be done in a newborn, while point of care test can be used in a newborn also. Now, for confirmation, even today, even after doing point of care test, we have to confirm by HPLC. We do molecular diagnosis, of course, is the best, but we cannot afford to do in all. Hence, we do for prenatal diagnosis, which is a must. Now, whom all to screen for sickle cell disease? Ideal is the whole population. However, if we cannot do, we must do newborn screening, antenatal screening, and prenatal diagnosis. I think these 3 are a major challenge which we face in our hospital is we cannot use any of this test after a patient has received transfusion. And most of the patients, they present with anemia at periphery and they are given transfusion, and when sent to us, we cannot perform any test for 3 months. So many of them are lost because the blood is not withdrawn before giving a transfusion. Next, please. Next slide. Okay, we know this is a very figure which we know that there is, of course, cure for sickle cell disease, which we cannot afford and which Dr. Shahji will be talking on. But the best way is to prevent sickle cell disease, and it is an autosomal recessive. So we have to screen all reproductive age patients, students, and before their marriage, to whether they are trait or normal or SS. And one message we give is trait not to marry a trait. If they marry, then we have option for doing prenatal diagnosis. And with every pregnancy, there are 25% chance that the child can be a sufferer, a homozygous. Next, please. This is difficult. Theoretically looks very good, but it is difficult telling that don't marry a trait. Now, another thing I want to tell is we have to have newborn screening program in place, and we need very good test for doing newborn screening. And the sample used can be a cord blood, a Hill prick. They are simple. It can be taken on a filter paper and done. And HPLC is, of course, and molecular diagnosis is the confirmatory test. However, there are problems. We just screen and then leave the homozygous. We cannot do that. The most challenge is when we diagnose, assess newborn, we have to follow them up and give them comprehensive health care. Next, please. Now, these are at the end of now, after 40 years, what we have at medical college is everything except bone marrow transplant, and we are offering everything to them. Next, please. I think most of the centers in India should have this. Next, please. Next slide. Next. Yeah. Now, these are the things which a patient needs. It's not that one time you give something. With every year, the treatment, the screening, the anticipatory guidance for sickle cell patient changes. And it is tailor-made. It may not be same for all patients. So there is a challenge here, and one has to have experience of what to do when. Next, please. But I would like to stress few things. Now our patients are surviving, and most of them are in second decade. And what we see now is the multi-organ failure in them. And now to prevent them, to prevent stroke, we have transcranial Doppler. If it is abnormal, we put the patient on hydroxyurea and chronic transfusion therapy. Other very common feature in our part of the world is renal failure. We don't see much of pulmonary hypertension, which is seen in other patients, other part of the world. My patient's organ most commonest is kidney. So what we do is from the age of 5 years, we do microalbuminuria, and if abnormal, we put the patient on losartan. So it prevents or minimizes the involvement of kidney, pulmonary function test, and also fundus examination, because now we have laser therapy and we must look for sickle retinopathy, which we can treat. Next. And there are many other things like AVN. Next, please. Avascular necrosis, polylithiasis. Number of presentations differ according to the age which we see there, which I think I cannot present today here. The manage is mainly, of course, supportive, importance of hydration, balanced diet, sleep. They are really important. Symptomatic management, pain, transfusion. Preventive, I told you about how we have to anticipate complications, involvement of organ, and detect and treat them. Next, please. Next. Yeah. One message I would like to, I'm not going into the details of management of sickle cell disease, but one thing is one has to treat pain as a disease and not as a symptom. It's very important, and things have changed over a period of time, and we need to stress a lot, because every episode of pain, there is an organ being damaged, which we cannot see. Next, please. The other thing which I want to stress upon is the inappropriate use of blood transfusions. I think when I was a resident sickle patient, first thing we give is blood. I think that causes more harm than good. There are 3 indications for transfusion, where if you do not give, that we may lose the patient. But otherwise, unnecessary, like giving transfusion for a low hemoglobin. Now, hemoglobin is no more a criteria to give blood transfusion. Look at the clinical condition, and then because it increases viscosity and may precipitate acute chest syndrome, may precipitate stroke, which are very, very severe morbid conditions. Next, please. Next slide. Okay. Now, these are few important things which my patients must have this kit: a thermometer. They do not know bacha bukhar hai, nahi hai, a mask, a pulse ox, a tablet, water bottle, always, and a marker pen, a water resistant marker pen to mark the size of the spleen, to know if the patient is going for sequestration crisis, and a sickle diary for pain. Next, please. Next slide. Okay. Now, there are few drugs which are now disease modifying agents till now were hydroxyurea and blood transfusion. However, hydroxyurea is really a magical drug used for sickle cell disease, but it needs very strict monitoring, and which is difficult in our place. So hydroxyurea really changed the morbidity picture, and we have to see whom to give and how much to give. The other drugs which have come in last 3 years are crizanlizumab, voxelotrol, and L-butamol. Next, please. There are few innovations which we have to make for our country, maybe in the diagnostic test, in neonatal screening, hydroxyurea, novel way of counseling, we give cocktail. There are many things which we have to do. And we have brought out many policies from what we have done. Like, all sickle cell patients have free investigations. The government pays for their treatment investigations. They have a travel concession. They get monthly incentives. Different states give different incentive. Our state gives 1,000. Student-friendly rules. They get 1 hour extra in the exams. And now they've come under the Disability Act. Next, please. Next. Curative options. Of course, Dr. Shahji is the authority in that, and he'll be talking on that. Next. Next. Okay. Now, if you see a previous one, please, you see there are many guidelines you see in other countries. They are all evidence-based. But for India, we do not have such guidelines for our country. And we had one by NHM in 2016, which were mainly extrapolated from the other countries, which is not true. Our sickle cell is different. Malaria is common in sickle cell disease. Why they say they are protected against malaria? There are many other things. And now we are coming up probably with a good manual for sickle cell disease, which is yet to be released, and that would be helpful, I think. Next, please. Okay, I think what we want for prenatal diagnosis now is fetal cells to be separated from maternal blood instead of doing this invasive procedures. If we, I think that must be, you all must be trying that. I'm not aware of that, but we should be having that soon. Next, please. I think this was 2006 when we had a policy for Maharashtra state, and sickle cell program was launched in our state. Thank you for patient listening. Thank you so much.
Thank you. Thank you, Dr. Dipty Jain, for very informative and practical information about sickle cell, and many things we were not aware, although it's common. What we will do is we will have the next presentation, and then question and answers in one go. Next speaker is Dr. R.V. Shaji. Dr. Saji received his doctorate from Christian Medical College, Vellore in 2001, and has held postdoctoral fellowship from St. Jude Children's Research Hospital in Memphis at the University of Chicago. He is currently working as a professor at the Department of Hematology in CMC Vellore, and he is also adjunct scientist at the Center for Stem Cell Research, a unit of INSTEM at the CMC Vellore, where he runs the gene regulation lab. And his research interests primarily lie in the disease modeling using induced pluripotent cells. Dr. Saji's lab succeeded in reprogramming cells drawn from adult mice and making them function like stem cells around the human embryo, paving the way for a new chapter in stem cell therapy in the country. Such approaches can be used to study genetic disorders relating to blood, muscle, brain, and even disease like diabetes. The Latsaji is a very reputed active scientist in this very important area, and had several occasions to listen to him. A very impressive work and very intense researcher. I request Saji to make his presentation, and then we can go for discussion.
I first thank the organizers, uh, Vasant and Dr. Rakesh Mishra, for inviting me to give this talk. As Dr. Dipty Jain mentioned many times, one of the major areas that I have been working for a long time has been hemoglobinopathies. It's a very passionate field where I have a genetics lab where I do the genetic diagnosis for hemoglobinopathies. But there was a demand for doing basic science at CSCR, Center for Stem Cell Research, which is a unit of INSTEM, where I started induced pluripotent stem cells for studying hematological diseases. And I know that we don't have to give an introduction for induced pluripotent stem cells. If there are a few who do not know what induced pluripotent stem cells are, from fibroblast or any adult cells, it is possible to convert those cells to generate pluripotent stem cells. And these pluripotent stem cells have the properties of embryonic stem cells. And these, because of the properties of embryonic stem cells, they can be differentiated to multiple cell types, and therefore they are valuable tools for clinical applications and also for disease modeling. If we get fibroblasts or any other adult cells from the patients um, with complicated diseases, we can generate iPSCs, and they can be differentiated to blood cells, muscle cells, any cells, many cell types can be generated. And once this was shown by Shinya Yamanaka that it's possible to make induced pluripotent stem cells, the applications were very clear. From a patient, skin biopsy can be obtained, patient-specific iPSCs can be generated, and they can be repaired, and they can be differentiated, and they can be potentially used for transplantations. And there are few clinical trials which are successful now that these differentiated cells can be transplanted into the patients. And these iPSCs, they can be in vitro differentiated. And because these are derived from the patients, they will have the phenotypes of the differentiated cells, suppose neurons, will be defective neurons, and they can be used for drug screening. And therefore, it's very valuable tool, and these cells can be used for massive drug screening, and it's possible to identify the drugs, and they can be used effectively to treat the patient. And for disease modeling, the key point is that it is possible to get a continuous supply of the differentiated cells for disease modeling. I work on hematological diseases, and I work on extremely rare diseases, and I can get a maximum of 10 ml blood from the patient, and that is just not sufficient for studying the disease mechanisms, how the disease occurred, what mechanisms involved. With 10 ml of blood sample, it's absolutely impossible. But if I make iPSCs, the iPSCs can be differentiated to hematopoietic cells. These iPSCs can be banked in liquid nitrogen. Even after 5 years, I can take those iPSCs and do the experiments to study the disease mechanisms and for studying the pathogenesis of the disease. Conventionally, it's always start from a patient. It's possible to reprogram and make iPSCs, and they can be differentiated to different cell types, as I said, and then look at the phenotypes of the differentiated cells derived from the patient and the healthy individual and see what is the difference in the phenotype. But there is a lot of issues here. It's because of the two iPSCs itself derived from the same individuals, they can show a difference in the differentiation potential. And it's very difficult to see the subtle difference in the phenotype to say this is because of the disease. And also because of the genetic background of the patient and normal controls, there can be a background noise. And for getting sufficient information to know whether disease is mimicked, we have to generate iPSCs from multiple patients and multiple healthy individuals. Only then we can be confident that the phenotype that we see, it is because of the disease. However, with gene editing, it is possible that we can create mutations in the specific genes which are defective in the patients, and that can be done with the CRISPR-Cas9, and CRISPR-Cas9 can effectively create indels and disrupt the gene function. And then you know that this is the gene that is defective. These mutant iPSCs can be used as an isogenic control. So you have a normal without any CRISPR-Cas9 induced mutation, then you have a mutant iPSC line in which you created the mutations. The remaining genome, all other sequences essentially will be the same, and you can do a better disease mimicking. And recent studies have shown that you can do DNA-based editing and you can create specific substitutions. While CRISPR-Cas9 creates indels, base editing can create specific substitutions. And therefore, we can create the mutant iPSCs, and the differentiated cells from a mutant iPSCs and normal individuals can be efficiently compared. And for this, one key reagent that we made in the labs, a lot of hard work that Vasant did when he was a member of our lab, and he generated an iPSC line in which the Cas9 is expressed on the AVS1 site, and we can induce the Cas9 expression when doxycycline is added in the medium, and it creates the indels very, very efficiently within a few days, and we could create mutations in several genes. A few of them I will explain. This is another protocol we established in the lab. If we have to do disease mimicking, then we have to differentiate iPSCs to hematopoietic cells. This again, Vasant was part of the team to develop this protocol where iPSCs were differentiated to hematopoietic cells. It's a very difficult protocol, and we could successfully generate this hematopoietic cells, and this hematopoietic cells could make hematopoietic colonies. I'm going a little fast because I just want to tell that what are the steps that one has to take for establishing a lab for disease mimicking using induced pluripotent stem cells. It's a very efficient protocol that we established. And Vasant did more experiments, and where he found that between the HSPCs formed on different days, they have typical characteristics, that they have differences. And he also used a small molecule to improve the hematopoietic differentiation. And he could get the HSPCs made with the small molecules. They showed better colony formation, and overall the efficiency of the hematopoietic cells formed increased further. And these HSPCs could be differentiated to erythroid cells. Now we have the protocol to generate the mutations in the target genes. We have the protocol to make HSCs, hematopoietic cells, and also to differentiate them to erythroid cells. And it took a long time for us to have all the protocols in the lab. And we have done RNA sequencing and confirmed that the type of HSPCs formed, they have specific RNA signatures. And this is all important, and subsequently they found that the HSPCs that we collected on one particular day of differentiation, they had better erythroid differentiation. The Sonam in my lab, she further improved this protocol by transducing with 5 transcription factors in the hematopoietic cells that are generated, and subsequently that she differentiated. This is from a Josdale's paper, and he had used this protocol, and we modified this by transducing the iPSCs with the 5 factors, and we could generate large quantities of better HSPCs, and we are using this protocol for studying the disease modeling now. And you can see the significant increase in the expansion of the HSPCs when 5 factors are added, and we are fully geared to use these protocols and the reagents for studying the disease mechanisms of for rare hematological diseases. And just to go in the details of the diseases that we have been working on, and the 2 diseases, erythroid diseases, are congenital dyserythropoietic anemia and a diamond blackfan anemia. There is specific genetics, and specific genes are mutated, and the phenotypes both affect erythroid cells alone, and the genes have been identified And this is the most common mutations are these mutations. And when we do not know actual frequency, the prevalence of these diseases, and when we look at the websites, what we see is that a total of 200 to 300 patients have been reported in PubMed. But when you are in CMC, Vellore, we see at least 2 patients with the diamond blackfin anemia every month. And we get equal around 2 to 3 patients with congenital dyserythropoietic anemia, genetically characterized CDA patients every month. So it is not rare for a tertiary hospital like ours. And it is important to, because they're rare, it's there won't be a lot of effort to take the cells and study the disease mechanism and to understand how the disease occurred. And it is, for us, these diseases are not very rare. Just to tell about that brief introduction about CDA, it's a congenital dyserythropoietic anemia. Type 1, it's an autosomal recessive. And like I said, it causes ineffective erythropoiesis, and it has typical chromatin structures found in the erythroblast, and the mutations occur in a specific gene called codanin. And iPSCs generated from these patients can be kept in the liquid nitrogen. Whenever required, they can be differentiated to HSPCs. They can further differentiate to erythroid cells. Large quantities of erythroid cells can be generated in the lab, and they can be used for studying the disease mechanism. For doing this, what we did was we did not go and take the patient's cells. The reason is that by the time we get this patient cells and to create this iPSCs, it becomes very, very expensive. What we did was we took a wild type iPSCs and we used a modified base editing protocol and created specific mutations in the codanin 1 gene. And we could succeed with 100% efficiency to create these mutations in the specific region of codanin 1 gene. And subsequently, NGS and Sanger sequencing confirmed that we could get more than 95% of the base conversion. And one advantage of this is, unlike any other protocol, you do not have to do single cell sorting because we get more than 90% bases converted, and you can get predominantly mutant cells. And this wild type and mutant cells can be treated as isogenic, and they can be differentiated to HSPCs and erythroid cells. And such efficiency of base editing has not been reported, and we are very excited with this protocol that helps us to convert more than 95% of specific nucleotide in the target gene. And just to tell you an example, there's a codanin 1 gene where we could get nearly 100% conversion to create the mutation, creating homozygous mutations. And we isolate the clones and confirm that these mutations occur in almost all clones. And this is the obtained gRNA transmit. The target conversion efficiency is more than 90%, and this is not reported earlier. And several mutations we could create, and this was a gRNA with a very low efficiency where we could get heterozygous mutations. Here with high efficiency, we could get 100% conversion, and it was with, you can see here that 100% conversion could be obtained in the target sequence. And this is to just show you that almost all targets that we carefully designed the gRNA for, we could get very good conversion to create the mutations here. Now, what happened here is that we started with a normal iPSC line and created the mutations. We did not go and take patient samples and creating several iPSC lines. We saved a lot of time and saved a lot of money. And we differentiated them into HSPCs. And this is a specific pattern of expression of the markers that we see here. CD45, 235, you can see here in the mutant cells have a specific expression of these markers. And when we do the colony formation assays, you can see here the mutant cells have a different colony formation ability. They do not make efficient number of erythroid cells. We could successfully mimic this erythropoietic defect in the cells. And this is exciting because we do not have to go to patient samples to generate these iPSCs. And we are we have generated a lot of data, and we're trying to create more mutations. We have done the liquid culture. In the liquid culture, we could see the specific chromatin structures of in the erythroid cells that clearly shows that the disease is mimicked. And now we are doing further experiments using CRISPR gRNAs to revert the phenotype for disease correction. And other disease is diamond blackfan anemia. This is a disease that affects the ribosome biogenesis, and most important genes are this. And we know that this in the normal erythropoiesis goes on well. In DBA, what happens here is that after the when the HSC differentiates to make the BFUE between the blast forming unit and colony forming unit, there is a defect in the erythropoiesis. And this mutation does not affect any other cells in the body, it affects only the erythroid cells. And that is the key feature of this disease. And we have done NGS and specified the mutations here. We did erythroid culture and found that there is the arrest in the erythroid differentiation from these patients. And many experiments we have done to confirm that the patient samples that we used are for DBA. And as I said, what we did was we took the iPSCs, we tried to create mutations. And what we found was that both homozygous and heterozygous mutations prevented the survival of the iPSCs. The iPSCs could not survive at all. And we totally failed here. We thought that we would be able to make heterozygous and homozygous mutations. Then we realized that haploinsufficiency, that is heterozygous mutations in the ribosome biogenesis genes, that affects the survival of pluripotent stem cells. So we could not move forward in that one. We're doing other experiments to make sure that this is what is happening, and we have more data generated. I think we'll have more opportunity to talk about that. And this is totally unexpected data. Therefore, now we had to go back to the patients and sample. We created using Sendai viruses, we generated iPSCs, and what we found was that we could generate only 2 iPSC colonies, stable iPSC colonies from the patient. It clearly showed that haploinsufficiency of the RP genes, like we observed when we tried to create mutations, haploinsufficiency affects the pluripotent stem cell survival. And that clearly shows compared to the previous studies that creating generation of iPSCs from patients with DBA is difficult, and because the DBA genes are important for the reprogramming process to happen. So this is a disease, it's very complicated. We could not make a large number of iPSCs for disease mimicking by creating mutations or directly from the patients, but few iPSCs that we had it, and they're still in culture, and we're really confused that if RP genes haploinsufficiency is affecting the reprogramming, how did these 2 clones grow? And we are doing exome sequencing to identify whether they have additional mutations which helped these 2 clones to grow in culture. And there's a scientific reasons, because the patients which can accumulate somatic mutations in the hematopoietic cells, and they can show the phenotype reversion. We can go through the details of that if there are any questions there. And right now, what we're trying to do is that trying to create iPSCs from RPS and RPL genes, because there are 2 types of RP genes, the RPS and RPL, and it's not known why there is a difference between the RPS and RPL gene mutations. And we're trying to create mutations from both RPS and RPL, and trying to create a comparison of genotype and phenotype, trying to do exome sequencing in the iPSCs generated, and also in the total pool of PBMCs. And we have reasons, we're ready to answer those questions if there are any. And we have successfully generated lentiviral vectors which encode RPS19 gene, and that can be used for the gene therapy for diamond blackfan anemia. And the third disease, Vasant, do you have time? Sorry, it's not looking at the watch.
Yeah, go ahead. Yeah.
So this is a disease, the third disease, the Fanconi anemia. It's a DNA repair disease. And, we can see in the literature 1 in a million is the prevalence that's reported. However, what we see in CMC is more than 50 patients every year. And it's rare elsewhere, but in India, it is not rare at all. And since I started the study 3 years back, I could recruit more than 200 patients, new patients, and also the patients who were followed up in our department. And so it is rare. If you look at the total population, but for a hospital like ours, these patients are not rare. Fanconi anemia patients are not rare. And the mutations can occur any one of the 22 genes in the FA-DNA repair pathway, and they have typical phenotype, and one of them is progressive bone marrow failure, and that is a typical phenotype, and the bone marrow become aplastic, and they show pancytopenia at a median age of 7 years, and they have congenital malformations and increased risk of malignancies. And the most sensitive phenotype is that the patient cells are extremely sensitive to DNA cross-links. And what is interesting phenomenon that happens in the patients is 25% of the patients, there is somatic reversion and they become mosaic, and their hematopoietic cells alone get corrected. And that is because of many molecular phenomena that we know that happens in the HSCs. And this is to show it's a complicated pathway with this upstream complex, downstream complex. One of the key events that happens in this here is the ubiquitination of FANC-I and FANC-D2. And this is an important event that is monitored in the patients to see whether the patient has Fanconi anemia at all. And this patient's diagnosis is very important because it's very difficult to differentiate between the idiopathic aplastic anemia, which is an acquired disease, and Fanconi anemia. And we routinely do a Western blot of this FANC-D2 to see whether it's ubiquitinated or non-ubiquitinated. And the patient samples will show that lack of ubiquitination of FANC-D2. And we have done an extensive DNA that we have established a very good DNA diagnosis algorithm where we have a lab which our cytogenetics lab performed the chromosome breakage analysis to see whether this DNA breaks exist when the patient cells are treated with intestine cross-link agents. Intestine cross-link agents, when they get cross-linked with the DNA, and they will not be effectively removed by the patient's patient cells because of the active FA pathway. But the normal cells, the intestine crosslinks will be removed. When the intestine crosslinks are not removed, chromosomes will get weird structures and typical triradial formations, and that is a diagnosis test. And we also said, like I said in the previous slide, we do a FANC-D2 ubiquitination to see whether the patient has got defective FANC-D2 ubiquitination. To say it is FANC-D2, FANC-D2 is absent, the patient is fine. Again, there are several reasons why it is important to do accurate molecular diagnosis of Fanconi anemia. And we classified these patients whether they have upstream defect, downstream effect, or whether the patients have mosaicism. We classify all the patients. This is an old figure, 119, and we have performed more than 150 patients now, and this paper is just sent for publication. And now we have access to the patient's fibroblast, and the we in this project we also wanted to make iPSCs. There, as I said earlier, the fibroblast can be taken and they can be reprogrammed with 4 transcription factors. These iPSCs can be obtained, and as it's been already shown that the fibroblast in which FA pathway is defective cannot be reprogrammed efficiently. What we did was we used a lentiviral vector through which the wild copy, the normal copy of the gene is expressed in a doxycycline inducible fashion. And then after the iPSC is formed, we remove the doxycycline and see whether disease mimicking occurs in the iPSCs and the HSPCs formed from the iPSCs. And this is one of the old studies which we published and say that we could effectively make iPSCs from a patient with Fanconi anemia using an inducible complementation system. And these iPSCs mimicked all the features of hematopoietic stem cells. And definitely, these iPSCs are much, much better than fibroblasts or lymphoblast or cell lines, which are commonly used for studying disease mechanism of Fanconi anemia. And we have currently differentiated these iPSCs to HSPCs. Unfortunately, we do not see a significant difference in the phenotype because the type of cells that are formed from the iPSCs, they are not true hematopoietic stem cells to show the phenotype. And Sonam's 5-factor protocol, we are trying to apply it here so that we be able to get much better hematopoietic stem cells, which are transplantable. And we think that those HSPCs will show the phenotype. Only then we are confident, we'll be confident to say that the disease mimicking really occurred. And we have done this since iPSCs are mimicking the HSC phenotype. We thought we did an RNA sequencing, and we found that there are specific pathways that are downregulated and upregulated in the iPSCs itself. And one of them is TGF-beta signaling pathway that's been recently reported as an important pathway, which is dysregulated in hematopoietic stem cells from patients with the Fanconi anemia. And several reasons for doing this, because there's FA pathway genes, they have canonical and non-canonical functions. And we generated iPSCs not only for FANK-A, which I described in the previous slides, we generated iPSCs by inducible complementation for several other genes. So we got fibroblasts from the patients with mutations in all these genes, and we complemented using a doxycycline-induced vector, and we made iPSCs from them. And this is the last experiment that we did was that it's only 4 or 5 genes mutations, 5 genes mutations are common in the Indian population. If I want to go and create mutations in all the 22 genes, it's absolutely impossible that I can go. I have to wait for at least 20 years to get fibroblasts from patients with mutations in all these 22 genes. So what we thought was, like I described in the previous 2 diseases, we tried to create mutations in the genes by using dual tRNAs, lentiviral vectors, and after that we sorted the BFA positive cells which are transduced with these lentiviral vectors, and we use that for disease modeling. And what is interesting was that again we used Vasant's inducible Cas9 iPSCs, dual tRNA vectors, and created the mutations in the 22 genes, and one cell line with mutation FANK-A, second cell line with FANK-B, like that 22 different cell lines we generated. Very interesting finding that we found here, that we could see that many genes which were thought to be involved in the FA pathway affecting the cell survival, and we've carefully looked at the cell survival by looking at the percentage of the transducer cells. And what we found was that many of them, when the doxycycline is added to create the mutations, the BFP positive cells, transducer cells, disappear, saying that the mutant cells, they did not survive. This is FANK-E is an example. And overall, out of 22 genes that we studied, 17 genes showed rapid cell death, and others, the 5 genes which are considered as FA pathway genes, they do not show the phenotype. And it's intriguing, and why those genes do not show. And we're doing mouse transplantation experiments. We're taking CD34 cells from a normal individual and using a mini library, trying to create mutations in all these 22 genes. And this current experiments, we're transplanting into NBSCW mouse to see all these genes, whether they have any difference in the generation of bone marrow failure in the patient. It's possible that one gene causes much more severe phenotype than other. And we also believe that at least 5 of these genes which do not affect the survival of the iPSCs, they may not be really involved in the FA phenotype at all. And with that, I thank a lot of people involved in the study, and Kritika, Gaurav, and Sonam Rani, and Vasant was sort of a mentor for both Kritika and Sonam, and Kirti Anurag and Tabanchan, who joined later with the new students in the lab. Anindra is a clinical fellow working with me, and he's did a lot of work for us, and the technicians and the collaborators. George, I should mention, is very passionate clinician, and sometimes he's more excited than me or my students, and he's one of the rarest clinicians that you see. Is very excited to know what happens in the lab. And my juniors in the clinical side, Fauzia, and from the lab at CSAR, many people, including Dr. Alok Srivastava, and a lot of support from the CSAR core facility and funding from Department of Biotechnology and DBT Welcome Alliance. Thank you for your attention.
Thank you. Thank you, Dr. Saji, for very informative, elaborate talk. Vasant, you will take over the question answer session. Yeah, please go ahead.
Hello, everyone. So I'm Vasant, and I lead the stem cell group at TIGS. So I would like to thank our eminent speakers for covering up a range of topics on epidemiology and screening to clinical intervention for sickle cell disease, and also how stem cell based disease model can be used to study disease pathogenesis in inherited hematological disorders, most of which that are yet to be well investigated in the Indian context. Moving on to the panel discussion. Sickle cell disease mutation has also been found to coexist with other hemoglobinopathies like beta thalassemia. So I request Dr. Jain to briefly tell us about the prevalence of sickle beta thalassemia in India and how patients respond to conventional clinical interventions.
Okay. Now, sickle cell disease is a syndrome. It's a sickle cell syndrome. Yeah. And we get many, what we call them as double heterozygous. So the commonest is, of course, sickle thalassemia, what we see, sickle beta thalassemia. And actually, it varies from my experience, it varies from region to region. Like in Nagpur, it is around 10 to 12% have double heterozygous sickle cell. That is, they are sickle thal. When you go to Akola, which we have reported also with Dr. Sargent, that it was around 30% had sickle thal. So it's varying from region to region. Areas which have high thalassemia population, there we see more of sickle thal. The other we see is SD, Punjab, D Punjab, and SE. We rarely see SC, which is very common in Brazil, States, and Africa. We rarely see SC, sickle cell disease. And that's why the phenotype is also different. Because SC is have more of ophthalmic and other things, and we have different double heterozygous.
Okay. Thank you, ma'am. And the next question is, even though the prevalence of hemoglobinopathies in India is well investigated, the same may not be applicable to other disorders like inherited bone marrow failure that Dr. Shaji was discussing now. So I request Dr. Shaji to briefly tell us about the prevalence of these inherited bone marrow failure syndromes in India.
Okay, Vasant, as I said, it's a very tough question. And because when I started on Fanconi anemia, I was inspired by Dr. Maman Chandi telling a clinical situation where a patient who was thought to have an aplastic anemia, and the patient was treated with a normal dose of drugs, and the patient died in front of his eyes. The patient had underlying Fanconi anemia, and then we started doing the diagnosis carefully, and we found that a lot of patients, young patients with aplastic anemia, they're not really idiopathic aplastic anemia. That is because of Fanconi anemia. And when we started doing the screening, we realized that we have at least 50 patients every year with Fanconi anemia. But that is much, much more than what is described in the literature. So I don't want to tell based on the literature. I can tell based on our experience here from patients who are referred from different centers that it is very, very high. And I have no idea what is the actual prevalence unless a study is done in the population.
Thank you.
Similarly, for the other two diseases, and I've been desperately trying to look at, I was expecting this question and I looked for it, and it says 200 to 300 cases reported in the literature. Okay. But that is not the situation here, what we see.
Okay. Thank you, Dr. So, Dr. Jain, you are one of the few clinicians who has worked extensively on sickle cell anemia in India for more than 3 decades. So, we would like you to recommend 3 key policy recommendations with regard to sickle cell anemia in India.
Okay. I think the recommendation I would have is first to have a newborn screening program, because I say Indian sickle cell disease is different from what we have in Africa and US. So from birth, we must know the sickle cell disease, the morbidity, what is their presentation, first thing. Second is our infection, the bacteremia, the etiology of infection is also different. I'm just giving you an example. What we are doing today is extrapolating the protocols from other countries, which are evidence-based in their country, to Indian population. I think it's time we need to revise this thing. And I think we must think, till now, even I was not thinking of anticipatory guidance and screening and even the option of stem cell transplant. I think this is high time. So many transplants have been done for sickle cell disease in our country, but hardly any from Indian population. We need to think about that.
Thank you, Dr. Jain. So, Dr. Shaji, being a pioneer in India in generating iPSC models to study RGDs, could you tell us how iPSC can enable the development of disease models for patient-specific clinical interventions?
Well, there are two studies where one of them is which is going on Parkinson's disease, and there is phase 1 trial that's going on. And the second one, which is the most successful, is for the eye diseases. Okay, and it's been shown that the differentiated cells could be successfully used for curing the disease. And there are no severe immunological complications at that site. Therefore, even embryonic stem cells were used earlier. Okay, but other diseases, I think we have to wait and see. For Parkinson's disease, there's a study. I mean, there's a clinical trial that's going on.
Okay. Thank you, Dr. So there are a few questions in the chat boxes. So I'll go through a few of them. So the first question is to Dr. Jain. So does SCD increases the likelihood of severe COVID-19 disease in children? If this does, can you shed light on the underlying mechanisms? So on sickle cell anemia and COVID.
Actually, it's a difficult question to answer, because during COVID period, sickle cell patients were not coming to the hospital, and mainly the experience I have is hospital based. They were asked, even we used to provide medicine at home, and it was mainly telemedicine, teleconsultation for them. However, from my experience, I would say that children had milder COVID. However, adults had moderate and severe COVID. And the mortality in adults in our sickle patients was more.
Thank you, Doctor. So the next question is to Dr. Shaji. So why does a defect in DNA repair, this is from Dr. Dashreddy. So why does a defect in DNA repair pathway lead to anemia? Or the erythroid progenitors are more prone to DNA damage? And what about other blood compartments like myeloid and lymphoid?
Right. So DNA repair disease is Fanconi anemia. It does not affect erythroid compartment alone. It causes pancytopenia. It affects almost all lineages, and the total number of hematopoietic stem cells all itself will be very, very, very few, like you see in aplastic anemia. And there are several reasons because of um, the Fanconi. When the intestine crosslinks are not removed, the DNA replication will be arrested, and there will be the consequences. There are many, and there will be increased ROS production, and the hematopoietic stem cells, they cannot replicate very well within the cells. Overall, the total number of cells will be very, very few. And if we start talking about the canonical and non-canonical functions of FA pathway, there is autophagy affected, mitophagy affected, and increased ROS, and there are several other factors.
Thank you, Dr. Shaji. So With this, I would like to thank our speakers once again. And the information provided will not only help for the general audience, but it will also help us at TIGS, where we work on stem cell based platforms to develop iPSC models for disorders like that are associating lysosomes and bone and muscle development, and also to treat blood disorders like hemoglobinopathies. So thank you everyone for joining us for today's webinar.
Thank you for the invitation.
Thank you.
Thank you, Vatan. Thank you, all the speakers, Dr. Jain, Dr. Saji, for giving us the most latest information about, rare genetic disease is a unique thing. And as we see that even Indian versions of sickle cell anemia is unique. So we have to solve our problems. And I think both of you are leading the way. And I hope that there is a hope now, as clearly we have seen from the talk, there are ways. And I look forward to seeing more such progress, and hope that we'll be able to contribute from our institute in this important problem that our society is facing. Thank you all participants for your questions and patient listening. And thank you all of you. Thank you. Bye-bye. Take care. Bye.
Thank you. Thank you.